Hello and welcome to Sciencera. In this video, we're going to discuss structures of proteins. I believe this video will be helpful for students who are writing biochemistry exam on 24th of May. Let's begin. Structures of proteins. Peptides and protein have 3D shapes or structures. Since the simplest dipeptide contain many atoms, it would be impossible to predict its shapes by VSEPR, as was done with simple organic molecules. Proteins have four levels of structures: primary structure, secondary structure, tertiary, and quaternary. Before we move on, please subscribe to my channel. You can also join our Telegram group by clicking on the link on the screen. Welcome to my channel Science Era. For anyone who are new to it, I make video every twice a week. Each level of protein structure is a result of interaction between amino acids of the protein. First is primary structure. Primary structure is the order in which the amino acids are joined together by peptide bonds that form the backbone from N terminus to the C terminus. The amino acid chains are subsequent to this backbone. The order or the sequence of amino acid in a protein chain are important in determining its structure and function. Arranging amino acids in a different order creates a polypeptide or protein that no longer has the same function as the initial sequence of amino acid. For example, 8 amino acid peptide angiotensin 2, which is involved in normal blood pressure regulation in human, has the following amino acid sequence. Any other order of the amino acid in this peptide would result in a peptide that would not function as angiotensin 2. Then we have secondary structures. The secondary structure of protein describes repeating pattern of structure with 3D structures of the protein. Two bonds are involved in secondary structure are hydrogen and disulfide bond. The two most common secondary structures are alpha helix and beta-pleated sheet. The alpha helix is a coiled structure, and much like the coil of the telephone cord, it is a right-handed coil. This coil is stabilized by hydrogen bond between carbonyl oxygen of one amino acid and nitrogen hydrogen bonds or atoms of another amino acid located 4 amino acid from its the primary structure. The coil is able to stretch and recoil and is a strong structure. The side chain projects outward from the axis of the helix. You can see on the screen a secondary structure involving the hydrogen bonding along the backbone. Here you can see the beta-pleated sheet. The beta-pleated sheet is an extended structure in which segments of protein chain align to form a zigzag structure as given on the screen. You can see this is labeled as N terminus and this is the C terminus and the hydrogen bond between peptide backbone. Here you can see the parallel and anti-parallel chains of the protein. Strands called beta strands are held together through hydrogen bonding interaction between the backbone. The side chain of a beta-pleated sheet extend above and below the sheet. Glycine, serine, collagen are all the examples of protein with beta strands. Then we have third type of structure, tertiary structure. The interaction of a side chain within the secondary structure lead to the tertiary structures of the protein. The tertiary structure is the three-dimensional structure of the protein. It involves twisting and folding of polypeptide chain caused by hydrophobic and hydrophilic interaction between the side chain of amino acids. The non-polar amino acid side chain and end up the non-polar amino sides chain end up in the interior of the protein away from the aqueous environment. The polar side chain appear on the surface of the protein since they are attracted to the aqueous surrounding. Stabilization of the tertiary structure is by attractive force between the side chain and aqueous environment and the attractive force between the side chains themselves. These attraction forces cause the protein to fold into a specific 3D shape. Interaction in tertiary structures involves are nonpolar or hydrophobic interactions, polar or hydrophilic interaction, salt bridge also known as ionic interactions, and disulfide bonds, which are covalent bonds formed between the sulfur and hydrogen group of two cysteine molecules. Here again you can see the diagram of protein. You can see the hydrogen bonds. These are the disulfide bond, hydrophobic interaction, hydrogen bonds to water, which is hydrophilic interaction. Here you can see the salt bridge on the diagram. Here again one of the hydrogen bond. Proteins are classified based on their three-dimensional shape. Globular proteins are compact spherical structure that are soluble in an aqueous environment. Myoglobin, which stores oxygen in a muscle, is an example of globular protein. Then we have fibrous protein. Fibrous proteins are long thread-like structures that have high helical content. Keratin found in hair, nail, scales of reptile and collagen are all the examples of fibrous protein. Let's look at the three-dimensional structure of the protein. Quaternary structure. Quaternary structure is two or the more polypeptide chain interacting to form a biologically active protein. Hemoglobin and oxygen transport protein is an example of protein with a quaternary structure. It consists of four polypeptide chain or subunit. It has two identical alpha subunits and two identical beta subunits. All four subunits must be present for the protein to function as an oxygen carrier. So, hemoglobin is a quaternary structure. Summary of levels of structure and stabilizing force in the protein. Primary level of structure. Force stabilizing proteins are peptide bond. In secondary level of structure, forces stabilizing the structure of protein are hydrogen bonding along the protein backbone between amino acids close together in the sequence. Then we have tertiary structure and force stabilizing them are London forces, hydrogen bonding, dipole-dipole movement and ion dipole interaction, salt bridge and disulfide bonds between amino acids far away from each other in the sequence. Then lastly we have the quaternary level of structure. Forces are same as tertiary structure but between the subunits. Denaturation of protein. Denaturation is a process that disrupts secondary, tertiary, and quaternary structures. The primary structure is not destroyed during the denaturation. Frying an egg is an example of denaturation by heat. Heat disrupts the intermolecular force such as hydrogen bonding and polar interaction. Other denaturing agents are change in pH, which alters the ability of acidic and basic side chain to form the salt bridge, organic compound which will disturb the disulfide bonds, and mechanical agitation which disrupts the hydrogen bonds. Here you can see the example of protein denaturation. Denaturing agents are heat above 50 degrees Celsius. What will happen? Hydrogen bond and hydrophobic interaction will disrupt. Example is cooking of food. Acid base denaturing agent, salt bridge and hydrogen bonds will be disrupt. For example, in lactic acid from bacteria which denatures milk protein in preparation of yogurt and cheese. Third is organic compound, which also act as denaturing agent, which will disrupt the disulfide bonds. For example, thiols, which are used in hair styling for hair straightening and permanent waves. Then we have heavy metal ions. They will disrupt the disulfide bonds and salt bridges, for example, mercury and lead poisoning. Then we have mechanical agitation, which will also disturb hydrogen bonds and London forces, for example, whipped cream and margarine made from the egg white. You can see an example of denaturation of protein using egg. Here you can see the active protein in the egg. When it is heated or contact with acid base or heavy metal salt agitation, they will denature and the protein will change its shape. Curling straight hair or straightening curly hair requires protein denaturation. Both processes require disruption of disulfide bond found in the hair protein keratin. The disruption of disulfide bond reshapes the hair and forces the reformation of the disulfide bonds in a new place. Ammonium thioglycolate, which reduces or breaks disulfide bonds, and hydrogen peroxide, which reform the disulfide bonds, are the two chemical agents used in the hair styling. Process of denaturation is used as an antidote for lead or mercury poisoning. Egg whites can be given to an individual who has ingested a heavy metal. Egg whites are denatured by the heavy metal and a precipitate is formed. Vomiting is induced to eliminate the metal protein precipitate. Example of biologically important proteins. Number one is collagen. Collagen is the most abundant protein in the body. One third of body's protein is collagen. It is found in connective tissues like cartilages, skin, blood vessels, and tendons. A special quaternary structure called a triple helix forms the fibrous structure of collagen. Here you can see the structure of triple helix. Each polypeptide chain of a triple helix is a left-handed helix. Collagen primarily contains the amino acid glycine, proline, alanine, and hydroxyproline. Then we have hemoglobin. Hemoglobin transports oxygen in blood. It is composed of two alpha subunits and two beta subunits held together by a hydrogen bond, London forces, and salt bridges. Each subunit contains a non-protein part called a prosthetic group, which is called a heme. Each heme group binds to a ion which binds oxygen, so a molecule of hemoglobin can bind up to the four molecules of oxygen. When oxygen binds to the ion of the hemoglobin, the shape of hemoglobin changes, which allows the hemoglobin to hold onto the oxygen until it is delivered to the tissue. This type of change in shape is called the conformational change. After the oxygen is delivered to the tissue, the shape of hemoglobin changes back to its pre-oxygenated form. Here you can see on the screen, this is the deoxygenated state and this is a oxygenated state of hemoglobin. Antibodies. Antibodies are your body's defense system. Antibodies, also known as immunoglobulin, are produced in our bodies when a foreign agent like bacteria enters. The foreign agent recognized by antibodies is known as an antigen. Antibodies consist of four polypeptide chain held together by disulfide bond and intermolecular forces. Antibodies are Y-shaped. Antigen binds at the top of each arm of the Y shape. Antibodies, your body's defense system. The top of each Y has a unique primary structure for a particular antigen and binds only one antigen, which is then destroyed by the immune system. This brings us to the end of structures of protein. Thank you for watching. If you like my video, don't forget to subscribe for more.